Thank you for the organizers for asking me to give this presentation for this course. During my talk, I'll try to make it clear that although we work with osteoclasts in vitro, it will actually show in vivo relevant information, and that if you pay enough attention to the details and the numbers, you might actually learn quite a lot more than what you would think. So, since I do not know at what level we are all at regarding bone, I just want to briefly summarize that our bone is being turned over all the way through life, and that this is done through mainly 4 cells. One is the osteoclast, which degrades the bone. It's the reversal cells that clean the surface and prepare the bone for formation. It's the osteoblasts that actually do the bone formation by depositing new collagen and catalyzing the mineralization. And then it's the embedded osteoblasts that become osteocytes that continue to build and remodel the bone by also again depositing collagen and catalyzing mineralization. And in an histological section, you can see these 4 cell types. As you can see here, here's an osteoclast, here are the reversal cells, here are the osteoblasts, and here's an osteocyte. Now, in my talk, I will focus on the osteoclast. And as I said, the osteoclast resorbs bone, but it can be hard to imagine how the osteoclast actually removes this bone and what it looks like in vivo. Here you can see a cavity that is formed in vivo, and over here you can see another section with an osteoclast that has made a more shallow cavity. But what does it really look like in 3D? In order to help you visualize this, I have brought with me some images from scanning electron microscopy. These images I have made from bone of sheep that have osteoporosis. So after having removed all the cells, so we can look at the bone surface itself, and looking by the scanning electron microscope, I hope you can appreciate that here you see a zone which has been resorbed by most likely multiple osteoclasts. This bone surface here is untouched, while this is where the cavities are, and some of them are long and some of them are more round. In this image, you can see a lot more bone resorption, and in this area, you can see that there's lots of resorption going on top of each other. So here, multiple osteoclasts have resorbed the same area many times. But here you can see just single osteoclasts have resorbed just a single cavitation, which is long, and the same is taking place here. Down here you can see the same round resorption cavities as before, here like in a row, and ending in a long cavity again. And again here in a zoom in of a very heavily resorbed area, you can see that just before we took the biopsy, an osteoclast has just scooped through the surface and made a long resorption cavity. So this is what the cavities look like in vivo. Osteoclasts are multinucleated cells that form through fusion. Their purpose is to degrade bone, as I already said. And the mechanism by how it does this, or the rationale, the basic mechanism for how it resorbs bone is pretty well known. In this image here, in this cartoon, you can see that an osteoclast, in order to resorb bone, needs to make a sealing zone. And this sealing zone is made up of F-actin and podosomes, which firmly contacts and is linked to the bone surface, creating a sealed zone in which the osteoclast can release protons and proteases and collagenases that eventually will erode the bone and create a cavity underneath the osteoclast. Now, because of this firmly sealed zone, then the osteoclast, the model was through many years that the osteoclast has to remain immobile during resorption. And because it was thought that you could not have a tight sealing zone if the osteoclast would move at the same time. But is this really true? Now, it is indeed so that osteoclasts, when resorbing, also make actin rings, as shown here. And the actin ring and the resorptive cavity, or the ruffled border, you can see here in the rotating image. But it is also recognized that there are other osteoclasts that do not match the model up here, where the actin ring is not round, but rather crescent-shaped, and where the front part is a half moon, but that the back part, which is the back part of the sealing zone, actually falls within the resorption cavity. So is this really an important observation, or is this just like an in vitro artifact? So we have through years published and documented that osteoclasts do have two different resorption modes, one called pit mode and the other trench mode. But it was only in 2017 when we managed to do time-lapse recordings that we could actually document it. So we have done here, we have taken bone slices, we have coated them with rhodamin, so the bone surface becomes red labeled. The osteoclasts, the human osteoclasts, are labeled with C-actin, which labels the F-actin of the osteoclasts. And then we have done time-lapse recordings over 3 days. And in this video, you can see how an osteoclast makes two different resorption cavities. And what you can see if you pay attention is that the osteoclast makes a round actin ring, and the ring is firmly remains immobile. Then it's dissolved and the osteoclast makes a new actin ring. That matches very nicely the dogma that I just explained you, that osteoclasts are immobile during resorption. And it also matches the resorption cavities that we see in vivo. But we have also shown that osteoclasts can also move during resorption. This you can see in this recording. You can see how an osteoclast makes an elongated trench, as we have chosen to call them, while it moves. And you can also see that these resorption cavities that it makes are precisely the same dimensions and length. Well, you cannot see it, but you have to believe me. They are the same dimension and length as those we observe in vivo. So osteoclasts have two different resorption modes. That is clear. But does it really matter? Well, in order to address this, we actually started out in the beginning not thinking that there were two different resorption modes. And I investigated the effect of prednisolone on osteoclasts in vitro. And first, the conclusion was that there was no effect. There was no difference in resorptive activity as the total eroded surface. But I did notice that the type of resorption was different. The cavities have a different shape. And it was only when I started analyzing the surface again and then distinguishing between whether they were long and elongated or if they were round, that I did find that actually prednisolone induced by threefold the osteoclasts to resorb in the trench mode. And that since we have also shown that osteoclasts making trenches resorb faster than pits, and they are deeper than the ones making pits, then it is also a more aggressive resorption. And does this then also matter for in vivo? Well, we certainly think so, and we got a hint when we did a study where we tested osteoclasts from 13 males and 15 females, and the total eroded surface was actually not changed when you looked at it. But when you looked at the choice of the osteoclast, whether it makes a trench or it makes a pit, we could actually see that the male osteoclasts significantly more decided to make a trench than those from females. So this already showed that there is some kind of clinical interest to this. So we also tested the sensitivity of the two different resorption modes to a series of inhibitors. Odyncatip is a drug that maybe some of you will know. It was developed by Merck and came into phase 3 clinical trial, and it finished the trial, but because of a very few side effects of cardiovascular nature, it was abandoned. But it was a drug that, it is a drug that is targeting specifically cathepsin K. And here you can see that it specifically inhibits the trench-making osteoclasts, not those making pits. Another drug that has been used in the clinic for many years is zoledronic acid, bisphosphonate. And here we can show that it more strongly inhibits trench-making osteoclasts rather than pits. And we have tested in general a long range of inhibitors, and which all affect, most of them affect osteoclasts making pits and trenches differently. And this just goes to show that if one uses inhibitors and looks at osteoclast activity, it pays off to look at the details of the effects, because otherwise you may actually miss a significant result. So to summarize this part, osteoclasts have two different resorption modes: the drilling, pit-forming osteoclasts, which also represents a slow resorption mode, and the events are less shallow. This is in contrast to the bulldozer or the trench-forming osteoclast, which reflects a fast resorption and also deeper, and they are therefore also more aggressive, and they are differently sensitive to drugs. But these details are easily hidden in the numbers if you don't pay attention to the details. And paying attention to the details actually pays off. And that is what I'll tell you about now, because osteoclasts in the laboratory reveal important clinical and health-related information about the donor. Now I will tell you why I say so. So my PhD student, Anais Müller, who just finished her PhD last month, has done a series of experiments during her PhD using blood from 50 healthy female blood donors and looking at a series of different parameters. Her setup was so that she took the buffy coat from the blood donation, she isolated CD14 positive monocytes and differentiated them into osteoclasts using rank ligand and MCSF for 9 days. And then we got matured osteoclasts. These were then analyzed by nuclei for nuclei and number and etc. But they were also lifted and reseeded onto bone slices, where we could then analyze the bone resorption cavities by different means afterwards. The results came out as such that here you can see that the age of the donors were between 40 and 66 years of age, and that they were premenopausal women in green and postmenopausal in black. When we looked at the eroded surface, so the total eroded surface, we saw there was a more than 20-fold variation between the osteoclasts from different donors. And we also saw the same degree of variation when we just looked at trenches or just looked at pits. This big variation is a well-known phenomenon and is normally considered a huge problem for using human osteoclasts because it's interpreted as a problem with reproducibility of the assay. But is it really the assay that is imprecise, or does it really reflect individual differences between the donors? And this is what Anais has investigated in her PhD, namely to use this variation as a strength rather than a problem. So she has done many analyses, but one of the important ones is that she did statistical multiple linear regression analysis and coupled with likelihood ratio tests. She combined a series of in vivo independent variables and in vitro independent variables to test if they could explain the dependent variable, such as the total eroded surface per bone surface. And doing this, she found that age was a positive factor. So osteoclasts the older the donors were, the osteoclast became more active. The inverse was the case with P1 and P. There was an inverse correlation. Another way of looking at bone resorption in vitro is by looking at CTX in vitro, which is a fragment of collagen released by the osteoclast and which can be detected by ELISA. Then if we use this very common approach to measure activity, we did not get any significant results for in vivo parameters. But do note that we almost got significance for CTX in vivo. But on the one hand, this says that CTX seems not to be as a good parameter to reflect in vivo characteristics of the donors with the osteoclasts as does the total eroded surface. But it also still tells us that actually the whole resorption assay with in vitro osteoclasts does really reflect in vivo relevant parameters. This was strengthened even further if we just looked at trenches. So the trench surface per bone surface correlated strongly with age and P1 and P, so bone formation in vivo. But if we looked at those making pits, there was no correlation with the in vivo parameters at all. So it's the trench osteoclasts that reflect in vivo characteristics, not those making pits. And I can just add that we also found that years since menopause was also a positive factor had a positive influence on the resorptive activity of osteoclasts. Now, just very recently, we have started to look more elaborated on these data. So one thing is to look at whether the overall resorptive activity, what that is. But if we now instead look at what determines if an osteoclast decides to make a trench or a pit. And in order to look at that, we used the term trench surface per eroded surface. So how many % of the eroded surface was made up of trenches? Doing the same type of analyses, Anais found, surprisingly, that the total number of cigarettes smoked throughout life correlated positively with the osteoclasts making trenches. So the more cigarettes you had smoked throughout life, the more likely your osteoclasts are to make trenches. So that is really fascinating, and it means that osteoclasts from past or current smokers are more aggressive than those from non-smokers. And actually, we also have data that this also counts for in vivo. But now, the parameters that I have shown you up till now that correlate with resorption are all like age, bone formation, bone resorption, menopause, and smoking. And these are all parameters that are dynamic, that change through time. So how can it be that osteoclasts remember, or the monocytes can carry a message down to the osteoclasts that is the result of the differentiation? How can that be? It must be something with epigenetics. Therefore, I now have a master student, Salah Masmoudi, who is working on epigenome-wide association study on our admittedly small data set, but we are trying to expand it with more. But despite that it's a small data set, we already do have indications that actually DNA methylations already in the leukocytes could have predicted whether osteoclasts in culture made trenches or not. And he just today sent me these results showing that although there are not many, there are still some significant CpG sites with DNA methylation that are significantly correlated with the ability of osteoclasts to make trenches. Also, when correcting for the false discovery rate. So preliminary data show that DNA methylation may be a way to regulate osteoclast aggressiveness by switching on or off the trench mode. And analyses are ongoing, and they will be expanded and combined to also look at the effect of smoking, age, bone turnover in vivo, etc. So stay tuned for more results on this in the near future. And the summary is basically that osteoclasts in the laboratory reveal important clinical and health-related information about the donor. So thank you for your attention, and thank you for my present and past members of clinical cell biology, and for my national and international collaboration partners. Thank you.
Well, sure, I believe you